And welcome back to another video. Today we're going to be looking at homeostasis and specifically the case study of blood glucose regulation, and then at the end we'll be looking at how the blood glucose regulation can go wrong and loads of diseases such as diabetes type 1 and type 2. Okay, so let's get into it. First, we need to define homeostasis. And homeostasis is a state of balance among all the body systems needed for the body to survive and function. And we have two types of homeostasis. We have positive and negative feedback. So, negative feedback is when systems are restored to their original/optimal level, and positive feedback is when a system is taken away from its original and altered. Now, blood glucose regulation is in fact negative feedback body in the blood plasma. And if we have a low blood glucose concentration, this raises the water potential of the blood plasma. So water moves out by osmosis into nearby cells, for example, red blood cells commonly found in the blood, down its water potential gradient. This causes cells to swell and they may burst, and this is called lysis, and it's not good at all. You don't want this. On the other hand, you also don't want a high blood glucose concentration, as this lowers the water potential of the blood plasma. So, water moves out by osmosis out of nearby cells, like for example red blood cells again, down a water potential gradient, and this causes cells to shrivel up. Now, both having an extremely high and an extremely low blood glucose concentration is bad for the body, and this is why we have homeostasis in order to maintain this blood glucose concentration so it's at a healthy level. Right, so as I said, glucose is obviously travels around the body via blood plasma, and hormones can return the glucose concentration back to its optimum. Now, for a little quiz for you, is that positive or negative feedback? And the answer is negative feedback. Now, there's going to be three hormones that we're going to be looking at today. We're going to be looking at insulin, glucagon, and adrenaline. And these are all hormones that regulate blood glucose. So, insulin lowers the blood glucose concentration, and we're going to be looking at insulin now. Insulin is a protein made in the beta cells of islets of Langerhans in the pancreas, and they are released when blood glucose concentration is too high, and obviously insulin is there to lower it. Now, it targets mainly the liver and skeletal muscles, and it's important to note that although the insulin is made in the pancreas, no cells in the pancreas are targeted by insulin. As you can see, if we look at the diagram, we can see that the pancreas produces insulin, but there is no insulin targeting the pancreas. In fact, the high blood sugar that you can see there promotes an insulin release, and that's the arrows in red. Now, what does insulin do? When insulin binds to its receptor proteins on the cell surface membrane, vesicles with embedded glucose carrier proteins fuse with the cell surface membrane. Glucose carrier proteins allow glucose to enter the cell via facilitated diffusion, using a carrier protein obviously, and this activation of insulin receptors also activates enzymes that convert glucose to glycogen. Now, obviously glycogen can be stored inside the liver or even skeletal muscles if that is what it's targeting as well, and this namely decreases the blood glucose concentration and also allows us to store that glucose to be used for maybe respiration or other processes in the future. Now let's move on to glucagon. It's very important to note the difference between glucagon and glycogen. Glycogen is not a hormone, it is in fact a storage substance of glucose, and it is very important that we do not get those two mixed up. So glucagon is a protein made in the alpha cells of islets of Langerhans in the pancreas, so it's also made in the pancreas. And the point of glucagon is to increase the blood glucose concentration. So a low blood sugar or blood glucose will promote glucagon release from the pancreas, and it targets only the liver cells. So nothing else is targeted by glucagon, only the liver cells. Now, what does it do? Glucagon binds to the receptors on the cell surface membrane of the target liver cells. They respond by activating enzymes that convert glycogen to glucose and also convert glycerol or amino acids to glucose in order to increase the blood glucose concentration level. Now, this then moves out of carrier protein and moves out into the blood. The conversion of glycogen to glucose is called glucogenesis, and the conversion of glycerol/amino acids to glucose is called glyconeogenesis. Namely, another important term is glycogenolysis, which is the hydrolysis of glucagon to glucose, which is also used in the liver. Okay, so we've covered insulin and glucagon, as you can see on the diagram. Feel free to pause and have a look at the diagram. But the diagram does not show about adrenaline, which is a specific hormone used for specific situations. Now, it's produced in the adrenal gland and is produced during the specific situation of a fight or flight situation, and it's very different to glucagon in the way that it targets the skeleton as well as the liver, and it goes and does glucogenolysis, where it converts glucagon in the liver or in the skeleton, and it converts it to glucose to quickly increase the blood glucose concentration level. Now, this doesn't happen on a day-to-day basis. That's for glucagon, and it only happens during stressful situations when more glucose is needed to make ATP by respiration. Now, I couldn't find a proper image for the next process, which is called the second messenger system. I'm just going to talk it through, and you don't need to look at the picture because the picture has nothing to do with it. So, adrenaline binds to its complementary specific receptor site on the receptor protein, and this changes the shape of the tertiary structure, which then changes the shape of the adenylate cyclase's tertiary structure, which exposes the active site and activates that enzyme or that adenylate cyclase. And this converts ATP to cyclic AMP or cyclic adenine monophosphate. CMP binds to the protein kinase in the cytoplasm, and this activates the enzyme, which then activates another enzyme to hydrolyze glycogen to glucose. And this is the exact same for glucagon, and this is just entirely to increase the blood glucose concentration. Now, that is the second messenger system, and that's all we need to know. The reason why it's the second messenger is because the primary messenger is the hormone. Okay, so now for interest, we're going to learn about a bit of diabetes. So, type 1 diabetes is an autoimmune disease caused by the immune system destroying beta cells of the islets of Langerhans, and the pancreas can't produce insulin, and you need insulin injections in order to stay alive. Now, we can think about why having the pancreas not produce insulin could be an issue. And the issue is high blood glucose concentration. There would normally be a promotion of insulin release from the pancreas, but none of this insulin can be produced. So we end up having a high blood glucose concentration, which obviously goes and causes what we previously spoke about at the start, where we have a decrease in water potential. And this means that there is movement of water out of the cells into the blood. And this causes cells to shrink, and it's a major issue. So diabetic patients who have type 1 diabetes rely heavily on insulin injections in order to get that blood glucose concentration back down, because they can't produce their own insulin. Now, type 2 diabetes is slightly different, and is when the body has a reduced sensitivity to insulin, and it's commonly known in doctors that being obese or having an unhealthy lifestyle is likely to increase this chance of getting type 2 diabetes, unlike type 1 diabetes, which is genetical mostly. And basically, the body cells have reduced sensitivity to insulin. So when insulin is released from the pancreas and it goes to its targeting cell, instead of the insulin binding to the receptors, it instead can't bind to the receptors. So glucose stays in the blood, and there is a reduced sensitivity. So that does mean that some insulin does bind to the receptors, but there is a reduced sensitivity overall. So mainly most of the glucose stays in the blood and it's still at a high blood glucose concentration, which obviously causes the exact same issues. So I hope that video was useful, and I will see you in the next one.